And may I audible, sir? And if the
Yes, sir.
slides
Yes,
are
sir.
visible. Okay, okay, sir. Thank you. Thanks a lot, sir. So
Yes, sir.
today we shall be discussing on biodiesel drug delivery system, as the need of the hour is to develop the quality medications. As in COVID-19 pandemic, we have seen that we require a lot of medications, as day by day the diseases are increasing, and it is the need to develop the quality medications, and it is on the shoulders of the pharmacist or the medical persons, so that the quality medications may be available to the society. So in drug delivery devices, a lot of devices are available, but biodigestive drug delivery system or biodigestive drug delivery devices have their own role. Because in case of drug delivery systems, we have the system in which the drug is loaded, and the drug should reach in the systemic circulation or should be available to the body up to the mark, and it should remain for a long time, so the therapeutic effect may be up to the desired time. So in this context, the drug should be safe and should be available. But in case of conventional systems, there are some ups and downs in therapeutic index or therapeutic window. So again and again, we will have to take the medication, we will have to take the dosage forms. But in modified drug delivery systems and sustained release systems, we have seen that the therapeutic concentration can be maintained for prolonged time, and we need to take one or two medication dose within a day or within 24 hours. But the peculiar properties of bioadaptive drug delivery system is that these systems adhere to the biological membrane, and these systems remain in the vicinity of the biological membrane, so the drug is available to be absorbed through this biological barrier into the systemic circulation, and hence the stability of the drug is much more, and it is available for prolonged time. So this is the peculiar properties or characteristics of these systems. And as we can say, it is the system in which two materials, at least one should be the biological in nature and another one is our preparation, comes in contact to one another and they adhere. And after that, the drug is released to the systemic circulation. And the rationality behind that is that the formulation will be held on a biological surface for localized drug delivery, and the API is released close to the site of action with a consequent enhancement of viability. As the viability will increase, the effect will increase, as well as the stability will also increase, and much more amount of the drug shall be available for showing the therapeutic effect. And here, the purpose of drug delivery system is that drug carrier system, first of all, adheres to the biological tissue, and it increases the concentration gradient at the absorption site, and ultimately it increases the bioavailability. And that bioavailability of the drug is responsible for maximizing the effect or therapeutic effect of the drug. And these systems can also be used for local targeting to the mucous surface, as well as the maximum amount is available for absorption, and we can say that the drug will remain in the systemic circulation for prolonged time. So secondly, it will show the prolonged effect. And another one thing is that the drug will remain in therapeutic index for prolonged time, and the side effects can be minimized to an extent. In another way, biodegradation is the addition of a biological membrane to our drug delivery devices, and in mucoadhesion, it is the addition or the attachment of our drug delivery device to the mucus, as mucus is available in different routes of administration, and we can see in oral cavity we have oral mucosa, through rectal route, through vaginal route, through eyes, the mucus is available. So in these routes we can develop these type of preparations, and mucoadhesive dosage forms can be designed to enable prolonged retention at the site of application. As these delivery devices will remain for prolonged time at the site of application, so the availability of drug will be more, and these can control the rate of drug release and ultimately will improve the therapeutic outcome. Biodiesel.
Sorry to interrupt you, sir. Sir, excuse me, sir. Please make it a full screen, sir.
Okay, sir, I have made it full screen. You may see.
No, it is visible only small. Please, sir.
Okay, sir. I try from my side, sir.
Yes, sir.
Please.
Do the slide show, then it will actually come.
Okay, sir.
Yes, sir.
Is it
Yeah, now
visible?
it is. Yes,
Okay.
yes.
Thank you, sir. Thank you.
It's okay.
So biodiesel is the ability of a material to adhere to a biological tissue, and in case of mucodesal, the system of the drug delivery will adhere to the mucus, and that mucus may be available at different routes of administration, as we know. The ideal characteristics of biomucodesive approach is that the rapid adherence. So the drug delivery system should adhere firmly to the mucus or biological membrane so that it may remain in that contact for prolonged time. Up to what extent the drug delivery device will remain on that system, it will release the drug for prolonged time and ultimately will get more beneficial effect. And it will depend upon the physical properties as well as chemical properties or the appearance of our drug delivery devices. Biodegradable without producing any toxic byproducts. This is the thing, as in fabrication of our drug delivery devices, we use different polymers, and these polymers after degradation may provide the toxic byproducts. So this is the prime importance for selection of our polymer, that the biodegradable products of our polymer should be non-toxic. Otherwise, the goal of our drug delivery device will vanish. And in some cases, we can add the penetration enhancer, because these penetration enhancer will increase the penetration of API to the biological membrane, and ultimately this will increase the bioavailability of our drug. So these are some advantages of these drug delivery devices. These devices remains at the site of action for longer time, and ultimately API bioavailability is increased. And use of specific bioadhesive molecules allows the possible targeting of particular sites or tissues. As we know that some of the diseases can be cured by targeted drug delivery devices. As we know, the enteric-coated tablets or colon-targeted drug delivery devices are available. So if we want to modulate the release of drug from these devices, we can add these characteristics of mucodesal in colon-targeted as well as in enteric-coated preparations. To increase residence time combined with controlled API, it may lead to lower administration frequency, as the drug will remain for longer time in our circulatory system. So less frequency of dosing will be required to treat the disease. And dose-related side effects can be reduced as API localization will occur in the devices. Some of the disadvantages may be seen in these devices, because any drug delivery devices is also associated with some disadvantages of the shortcomings. So the turnover of mucin or the biological membrane diminish the adherence of these devices, and the effect of these may decrease. And another one thing is that these additions may create the irritation to the biological membranes. So these are some of the disadvantages of the system. And certain drugs, when ingested, undergo drug destruction. There are several drugs which are potentially in this category, and many of the drugs affect liver metabolism and also cause destruction by first pass metabolism of the other drugs. And in another case, oral injections may result in more exposure of a drug to the GI tract. One of the side effects of many antibiotics is the destruction of normal GI flora, and it may result also in destruction of beneficial microbes of our GI tract, and in these systems it may occur frequently. Mucoadhesive inner layer, that is called as mucosa inner epithelial cell lining, and it is covered with viscoelastic fluid, and this mucus is secreted by goblet cells, and the lines with epithelia by special exocrine glands with mucus cells, and this mucus is composed of water and mucin, and this mucin is anionic in nature. Due to this anionic nature, the cationic category preparation or those preparations that have charges on their surfaces, they have more potential to adhere to the mucous membrane. Other component that may be seen in mucosa, that these are mucopolysaccharide, protein, lipids, and electrolytes, and the thickness of mucus may vary from 40 to 50 micrometer up to 300 micrometer. And this is the physiology of mucous membrane. Here we can see the epithelial, lamina propria, mucolaris, mucosa, submucosa, and so on. And this is responsible for adherence of our drug delivery device. This is the composition, general composition of our mucus. Water, glycoproteins, mineral salts, and free proteins are also available in different compositions. And mucopolysaccharides contains up to 8 to 10% of monosaccharides, and these are L-fructose, galactose, N-acetyl D-glucosamine, N-acetyl D-galactosamine, and sialic acid. These sialic acid is responsible for anionic nature of mucus, and this is the structure of and the composition of the macromolecules of mucus. Oligosaccharides, cysteine residue, and repeated structure contains both of the proteinous structure of mucus contains COH and NH2 group here, and the ionization of the chains is responsible for the charges of mucus. And mucus layer covers the epithelial surface, and it plays various role: protective role, barrier, adhesion, and lubrication. And disulfide bonds are available for interaction of mucin, and the polysaccharides that are available on the surface of this one are responsible for hydrophilic bond formation with our bioadhesive or mucoadhesive drug delivery devices. And this is the structure of mucin. Here we may see the polypeptide backbone, polysaccharide side chains, and these side chains can entangle with the hydrophilic chains of our polymers that are available in mucoadhesive drug delivery devices. Several mechanisms of mucoadhesion have been described. So 7 mechanisms are available yet now, and since 1940, mucoadhesive drug delivery devices are in the market, and these are playing a major role. In mucoadhesion, the various steps have been described. Wetting and swelling of the polymer. Actually, this is the first step. When we administer the mucoadhesive drug delivery device, the polymer available in our device absorb the underlying water from the mucosa, and it swells. First of all, the primary step of the device is that it should retain water, and for that water miscible, or we can say those polymers that have the hydrophilic groups, that may be very good candidate for developing these devices. Second one step of this one is the interpenetration between the polymer chains and the mucosal membrane. And when these hydrophilic groups or hydrophilic chains come in contact with mucosa, these interpenetrate with the side chains of mucosa, And third one step is the formation of bond. That bond formation may be the hydrogen bond formation, van der Waals bond formation, ionic bond formation, or sometimes it may uh, be responsible for covalent bond formation. So here we may see that the device is going to swell and the chains are in the position to make the contact with underlying mucosa. And here the swelling of polymer occurs because the components of polymer have an affinity for water. In second step, the mucoadhesive polymer chain and the mucosal polymer chains intermingle and entangle to form adhesive bond. And the strength of the bond depends upon the degree of penetration of the two polymer groups. And in third step, the formation of weak chemical bonds between the entangled polymer chains occurs, and these bonds may be van der Waals forces, hydrogen bonds, ionic interaction may be there. So this contact between mucosa and to the surface of our drug delivery device is must. If the contact is very intimate, the adhesion will be very strong. If contact is not there, the drug delivery device may not adhere to the biological membrane. So on these basis, various theories have been developed to discuss the mechanism of bioadhesion or mucoadhesion. So electronic theory, wetting theory, adsorption theory, diffusion theory, fracture theory, mechanical cohesive theory, different theories have been narrated time to time. In electronic theory, has been proposed that both mucoadhesive and biological materials possess opposing electrical charges. If the uh, charge is available on our or on our drug delivery device, certainly it should form the electronic attraction or ionic attraction with our mucosa. And if there is any attraction, ultimately it will confirm the bond formation. So this theory reveals that when both materials come into contact, they transfer electrons, leading to the building of a double layer at, at the interface. And at that interface, attractive force within this electronic double layer determines the mucoadhesive strength. So this theory reveals the formation of double layer, and in wet theory, the formation of hydrophilic bonds, the retention of water. And in this theory, the polymers having the affinity with water, they create the bonds to underlying mucosa and firm and adhere firmly to the mucus. In this slide, we can see the contact angle, the role of the contact angle in adherence. If the contact angle is 0, then we can say it can spread on the mucous membrane firmly and adhere. So contact angle theory reveals that the adherence will depend upon contact of our polymers, or we can say our drug delivery device to the mucosa. In adsorption theory, the mucoadhesive device adheres to the mucosa by secondary chemical interactions, as we can see in adsorption, physical adsorption and chemical adsorption are there. If there are physical bonds and chemical bonds responsible for attraction, then these may create the interaction between mucus and our drug delivery.
ज्वाइन कर
Hello, sir. Am I audible?
Yes, sir. Yes,
Okay,
sir.
okay,
You are
sir.
audible.
Okay, okay. Thank you, sir. Actually, there was a network problem and I could not see on uh, the screen of my laptop. Actually, the slides were not slideshow. Just 1 minute, sir.
It's okay, sir. It's okay.
Am I audible, sir? Hello.
Yes, sir. You
Yes,
are
sir.
audible.
Yes, sir. Yes, sir.
Okay, thank you, sir. Thanks a lot. Actually, there was network problem. Actually, I could
Yeah,
not
yeah,
see on my screen, sir.
yeah.
So
Sir, just kindly the slideshow. Like right now, it is on not on the slideshow mode. Kindly do it on the
sir,
slideshow, sir.
please,
Yes,
see
yes.
this, sir. Okay,
Fine.
okay.
It's better.
Thank you,
Better, sir.
sir. Thank you. So in case of biodiesel polymers, the main thing is that our polymers should adhere to the biological membrane, and biological membrane or our mucous membrane have the enormous water. If the polymer is hydrophilic in nature, so it will have very good opportunity to be fabricated in mucoadhesive or bioadhesive drug delivery devices. So the bioadhesive polymers should be water soluble, and water insoluble polymers which are swellable, which may form the swellable network, can be converted into mucoadhesive drug delivery devices. And sometimes we require the cross-linking agents. As we know, in developing the microspheres or microcapsules, we require the cross-linking agent. For example, in sodium alginate, we add calcium chloride, and ultimately, or the bivalent or trivalent ions, it forms the beads. And these beads are cross-linked beads. When we add these beads in water, they swell down. If these are going to swell, then ultimately there are a lot of chances that they may form the bonding with mucous membrane or biological membrane. So the ideal characteristics of the ideal polymer is also there, that they should not form any toxic or non-absorbable byproduct or metabolite in our GIT, because the toxic metabolites or toxic component developed or formed after degradation of the polymer may cause the harmful effect in our body. Another one thing is that good spreadability. These polymers should have the interaction with mucous membrane as well as biological membrane, and if the spreadability is more, then more area can be covered by these, and the ultimate contact area or intimated contact area should be more, shall be more, and then we can say the bioavailability shall be more, because absorption area will increase. Wetting, swelling, solubility, and biodegradability properties also affect the selection of these polymers. Optimum molecular weight, large molecular weight polymers have good criteria, have good opportunity to be selected to be applied in the devices, and these should be non-irritant to the mucous membrane, because these devices may remain for prolonged time on mucous membrane or the biological membrane, and that contact time can create the irritation. So these should be free from any irritation, and these form a strong non-covalent bond with mucin epithelial cell surfaces, because without bonding, without adherence, the bioadhesion or mucoadhesion is impossible. These should adhere quickly to the moist tissue, and these should allow easy incorporation of the drug, because the drug entrapment or loading efficiency, if the loading efficiency or entrapment efficiency is low, then ultimately it will affect the release, and ultimately it will also affect the bioavailability of API. And not decompose on storage or during shelf life of dosage form. The polymer should not decompose during the storage as well as on the shelf life. And these should be approved by regulatory authorities. We have seen different drug delivery devices, and by different routes, the mucoadhesive drug delivery devices can be applied, and on skin, bioadhesive drug delivery devices in the form of patches can be developed. So rectal route, sublingual route, buccal route, nasal route, ocular route, or vaginal route, these can be administered for effective management of the disease. Sometimes we can add the penetration enhancer. The penetrating enhancer facilitate the permeation through mucosal membrane, and these can increase the bioavailability or the absorption, we can say, of the API. These would be safe and non-toxic, non-irritant and non-allergenic, and these penetrating enhancer may be of natural origin, or these may be of synthetic origin. These should be pharmacologically inactive as well as chemically inactive, and they should have no pharmacological activity within the body, and lot of the penetrating enhancers can be applied in developing these devices, as benzylsodium chloride, dextran sulfate, fatty acids, sodium EDTA. Some of the examples of penetrating enhancers are these. And lot of polymers, according to their sources, their nature, their molecular weight, have been categorized to develop these biodigestive drug delivery devices. Agarose, chitosan, gelatin, pectin, carboxymethylcellulose, halogenated carboxymethylcellulose, hydroxypropyl methylcellulose, carbopore, PVP, PVA, halogenated polymers, and so on. Number of polymers that have large molecular weight, those have good water retention property, good swelling, good spreadability have been applied in developing these devices. According to water solubility, CMC, sodium CMC, HPMC, MC, PVA, PVP, carbopol, acrylic acid, PEG, and so on have been applied in developing these devices. And according to the charges, aminodextran, chitosan, carbopol, sodium alginate, pectin, sodium CMC, and some of them are uncharged, like as starch, PEG, PVA, and so on, have been applied in developing different devices, and these have been used in developing tablets, layer tablets, and some of them have been applied in developing in situ gel, pH sensitive gel formation, and so on. According to the nature of the drug delivery devices, these can be categorized solid, semi-solid, liquid dosage form. Solid mucoadhesive dosage forms may be tablets, matrix, biodhesive microparticulate, biodhesive inserts, gels, ointments, film, patches, and in liquid suspension and gel forming liquids are available. For developing biodhesive tablets, we can use API, mucoadhesive polymer, penetrating enhancer, and so on. And these are the simple ingredients that we generally use in developing tablet preparation. And biodhesive tablets are generally immobilized drug delivery system because they adhere to the biological membrane and remain for a long time in intimate contact. During this time, the drug is released and ultimately enter into systemic circulation for enhancement of bio. Multi-layer tablets, partially coated monolithic tablets in the form of mucoadhesive tablets are available. And here we can see how we can develop the biodhesive tablets. So by using biodhesive layer and natural excipients and water permeable coating above the biodhesive membrane, we can develop these tablets in layer form, in multi-layer form, and these may have very good adhesivity to the biological or mucosal membrane. These are limitations of biodhesive tablets: small surface of contact with mucosa. Because as we know that these tablets will adhere to the underlying mucosa or underlying biological membrane, and that limited area will be for absorption of drug. And if retention of formulation is less, like in stomach, where the retention time is not more than 2 hours, the availability of formulation for absorption will be less, and ultimately it will affect the bioavailability of drug. Then lack of physical flexibility, and sometimes it is difficult to obtain high release rate, which is required for some drugs. So in this case, release is governed by different polymers. So these can modulate the release of drug, and ultimately the presence of drug in our systemic circulation will be less. But in some cases, drug is needed in our biological system to monitor, or we can say to control the disease. So in that cases, this cannot be the choice, cannot be the drug delivery devices or optimized drug delivery devices for treating the diseases. The extent and frequency may cause irritation to mucosa, and this may be the side effect of measurement. Beside this, we can check the swelling, we can assess the measurement of residence time, retention time, adhesive strength determination. In biopsy tablets, one of the technique is there, that is the scintigraphy technique. हेलो, हेलो सर, हेलो
हाँ सर, नमस्ते। मैं रविंद्र कुमार बोल रहा हूँ सर।
सर। सर नमस्कार सर,
सर,
एम आई ऑडिबल?
सर, yes, now you are audible.
Sir, due to network problem, I could not get the good connectivity with the participants, with the organizer, and sorry for
It's okay,
inconvenience,
sir.
sir.
Now you
It's
are
okay. It's
audible.
okay. Yeah.
Okay, okay, sir. May I continue,
Yes, yes,
sir?
yes, sir. Please, sir.
Actually, time is over. Within 5 to 10 minutes, I will
Yes.
complete my talk,
Yes,
sir.
sir. Yes, sir. Yes, sir. Yes.
Hello, sir. Am I audible?
Yes, sir. You are audible and presentation is also visible, sir. Kindly do on the slide share,
Okay,
sir, once.
okay, okay. Thank you, sir. Thank you for response, sir. So, sir, another way, sir, we can develop the amyloid acid gels for biodiesel purpose, and these gels can be formulated for buccal, nasal, vaginal, and rectal routes also. And in case of in situ gel formation, these are the solution form and formed gels upon administration to particular site due to phase transition in environment of that site. And nowadays in COVID-19, we have seen that WHO recommended the hand sanitizer. In hand sanitizer, we added carbopol 934 and 940, and according to that, we also added the triethanolamine. The triethanolamine increases the pH, and then carbopol 934 or 940 converts into gel formation, and that is due to the change in pH. So such type of pH change may occur at the site of action, and ultimately these forms will convert into gel. So this type of preparations are also available. The in situ gel forms can be used as a mucodation or biodation purpose. And sometimes the electrolyte composition can change the consistency of gels. And in formulation of biodiesel gels, we require the mucodhesive polymers, permeating enhancers, preservative solvent, and API. Thermosensitive gel forming systems. In these systems, uh, these can be developed from aqueous polymer solutions with temperature change, and thermosensitive polymers are also available for developing these, and these may be an isopropyl, acrylamide, poloxamer, and so on. In pH sensitive gel formulations, we can use the carbopol or polyacrylic acid, as these are pH sensitive polymers. Ion sensitive gel forming systems, we can use these systems, and these can be developed by which the solution of polymer containing drug triggered to gel form when contact with specific ion and specific site. And ion sensitive polymers, these may include sodium alginate, gelatin gum, glycoglucan, carrageenan, and these can be used for nasal, ophthalmic, oral, vaginal, rectal routes also. In sodium alginate, we have seen when we add sodium alginate with calcium chloride, sodium alginate, this person converts into hydrogel system, that is processing system. So such type of systems or ion sensitive systems are available for biodation or mucodation. And the evaluation of these gels or the quality control of these gels can be conducted by different parameters. These may be drug content, viscosity, pH, in vitro drug release. Addition properties can be determined as the methods described or adopted for the assessment of mucodation in tablets. Membrane viscosity, this parameter can be determined by the interaction between polymers and cell membranes. Electrical conductance can be used to assess the addition of these polymers to the biological membrane. Patches can also be developed in the form of transdermal patch as a bioadhesive delivery devices, and these are designed either for unidirectional release or bidirectional release, and use of impermeable backing layer will maximize the drug concentration gradient and prolong addition because the system is protected from saliva. And typically size of such polymeric patches may be 1 to 3 centimeters square, but may have dimensions as large as 10 to 15 centimeters square, and it depends upon the site of administration. And these biodhesive patches can be developed in such a way. These may have the backing membrane, drug excipient and adhesive layer, and mucosal membrane is there. And to this membrane, the adhesive polymer adheres, and the firmness or the contactness of these systems provide the modified release of drug, and the drug remains for prolonged time in our systemic circulation. These are the ingredients that are required in developing these buccal patches. The API mucoadhesive polymer, as the key role of mucoadhesive polymer is the attachment of these devices to biological membrane, and these are the parameters by which we can assess the performance of the formulation: thickness, uniformity, folding, endurance, uniformity of weight, drug content uniformity, and these are the very normal parameters, and these are also applied for other drug delivery devices also. The specific is one the adhesive strength. By using different methods, we can assess the adhesive strength of the preparation. Transdermal patches we can also develop as a mucoadhesive drug delivery device, as a bioadhesive drug delivery device, and thickness uniformity, moisture content, uniformity of weight, drug content uniformity, flatness study, surface pH, in vitro release, and adhesive strength. These parameters can be applied for evaluation of the preparation. Chewing-gums are also the preparation that comes under the category. And medicated chewing gum offers advantage in comparison to conventional oral, mucosal, and oral dosage forms, as local treatment of mouth diseases can be done by this preparation, and systemic effect after absorption through the buccal and sublingual mucosa or from the gastrointestinal tract can be modified. These are the components by which we can develop the chewing gums: gum base, bulk sweetener, softener, flavoring agents, coloring agents. By using these components, we can develop these preparations as a bioadhesive system. And these can also be manufactured by compression of powders or granules on a conventional tablet machine. And these are some of the drawbacks of chewing gum. The release into saliva and disappears rapidly from oral cavity because of involuntary swelling. So this is the drawback of this one, and this drawback can be seen in other preparations also. The concentration drug in the oral cavity always tends to decrease as a result of salivary dilution. So dilution of the drug in oral cavity ultimately affects the drug, and it is related to the bioavailability of drug in our systemic circulation. The drug is not protected from the environment found in the oral cavity. The nature of drug may be changed because the pH of oral cavity will certainly affect the ionization, and that ionization is responsible for absorption from oral cavity. Administration of such dosage form is restricted to short time periods because the delivery system in the oral cavity causes a disturbance in drinking, eating, and speaking. So we can say lot of preparations can be developed in the form of bioadhesive and mucoadhesive drug delivery devices. And the main intention to develop these preparations is that the preparation should remain in direct contact to the biological membrane, so that the drug may directly enter in our systemic circulation, and we may get the maximum therapeutic effect with lower side effects. So with these words, may I conclude this session. Here, sir. Hello. Hello.
Yes, sir.
Yes, sir.
Yes, sir.
Sir. So lot of preparations in biodigestive drug delivery devices are available, and